Welcome, and thank you for joining us for our live virtual MDA Engaged Community Education Seminar, where we are highlighting specialists in the Pacific Northwest today. My name is Nicole Petrowski, and I'm the Community Education Specialist for MDA. We appreciate you joining us virtually and are glad that we're able to share this important educational seminar for you. MDA is committed to community education. We believe in the power of bringing our community together for opportunities to learn from specialists and having opportunities to connect with one another. This event is part of our larger MDA Engage series with specific disease symposia and community education seminars and community webinars as well. Our community education seminars, which is what we are having today, are geared towards general neuromuscular disease topics. While each event will feature regional specialists covering these topics, each event will also feature a specialty topic, which you can view on the screen. These are the events we have lined up for the remainder of 2020. And our engaged disease-specific symposiums are geared towards a particular diagnoses, and topics include such as best practices in care, research, clinical trials, and more. To view agendas for these seminars and symposiums, as well as to register, please visit the MDA Engage section on mda.org. You will also find our past recorded events here as well for your viewing option. And we would not be able to put on our educational programs without the support of our generous sponsors. I would like to thank Numotion Foundation, Biogen, and Genentech for supporting this seminar. I would also like to thank their representatives who are in attendance today. Now I have a few housekeeping items to go over before we begin our seminar today. Just so everyone is aware, we are recording today's event and we will be posting it to mda.org's website for on-demand viewing in a few weeks, and to ensure that those who weren't able to join us live today have access to the information. For those of you joining our event live today, please know all phone lines have been muted. We will be having a Q&A session at the end of each presentation, so please utilize the Q&A icon to type in your question. If you look at the bottom of your screen, a tray of webinar icons are down there, and you simply click on the Q&A icon and type in your question to host. You don't need to wait until the presentation is over before submitting your questions. Feel free to type them in as they come up while you're watching. We also encourage participants to communicate with each other by utilizing the chat feature throughout the day. If you want to just say hi and let participants know where you're from, or you want to share a bit of your story with others, or if you'd like to obtain suggestions from others that are watching throughout the community, please use the chat feature. You simply click on that chat icon and make sure you click all panelists and attendees. That way the attendees who are joining us will be able to see your comments. And you just type that in. Feel free to use this throughout the day to connect with others. We do have people from all over the country, not just the Pacific Northwest, so we do have quite a vast audience today. You will notice also on the agenda that there are small breaks between the speakers. Feel free to stay connected to the live broadcast during these times. I will be setting up the next presenter, so you will hear our mic, just so you know. But if you want to get something to eat or grab a drink of water, feel free to go too. Finally, we will be sending out a brief survey afterwards, and we would like to receive your feedback on what you heard today. We want to make sure that we're discussing topics that are of interest to our audience, and your feedback is very important to us. So thank you in advance for taking about 5 minutes to complete that survey. So with those details out of the way, I am pleased to introduce to you our first presenter, Dr. Thomas Ragole from Oregon Health and Science University. He completed his neurology residence at the University of California, San Francisco, and his fellowship in neuromuscular medicine at Harvard Medical School, Brigham and Women's, Massachusetts General. He is currently assistant professor of neurology at Oregon Health and Science University. His work focuses on clinical trials and research on myasthenia gravis, ALS, and medical decision-making. Dr. Ragole, thank you for being here today, and I will turn the presentation over to you.
Hi there, everybody, and thank you very much, Nicole. I'm very happy to be here today. I am going to share my screen here in a second. Let's see what happens here. as always, even though we've done these Zoom and Ring meetings forever, there's always a little bit of technical difficulty. Let's see what happens here. So Nicole, can I share my screen to everybody? It says that you are doing, there we go.
Yep, there you go.
All right, now we're in it. Okay,
There
sorry
is a
about
little
that,
bit of a
everybody.
delay. There you go.
All right, let's get started. So again, thank you, Nicole. Thank you to the MDA for having me. What we're going to be talking about today is a smattering of research updates in muscular dystrophy and other conditions that are supported by the Muscular Dystrophy Association. So with that, we'll get started. All of us academics, we always have to have these disclosure slides. These are things that could conflict with what we're saying. So there's nothing that I have that's relevant to this, but I think it's really important to note that we're going to be talking about some experimental therapeutics, experimental techniques that have not been evaluated or approved by the FDA. So it's worthwhile to know that these are things that are out there, but have not yet been vetted by safety and regulatory agencies completely. I do a couple of pharmaceutical funded clinical trials here, and I have done some consulting in the past. So we'll move on to our outline. So this is really the focus of today. We're gonna start way back in the day, where we've been. We'll talk about the MDA's role in all of these developments. And then I'm going to give you some updates by a few different conditions. Then we'll talk about where we're going, what's next, how to get involved, and then we'll leave about 10 minutes at the end for questions. And I think it's worth mentioning up front that I'm going to leave a lot of things out. I know that there are folks out there who have loved ones or may themselves have one of these muscular dystrophy conditions or MDA supported conditions that I'm unfortunately not going to be able to mention. 20 years ago, I really think that in an hour, you could probably give a lot of the research updates about every condition. But today the science is really just far too complicated, far too advanced. There's too many conditions that I think it's even possible to cover. And I put this picture of all these galaxies from the Hubble Space Telescope because my little brother is a physicist and he likes to remind me how much more complicated his job is than mine. And I think that this is a good example of just how complex, and how advanced our science has become. And really, I think that what we should keep in mind as we go forward is that this is really just a sample of what's possible. So we'll start way back at the very beginning, and I think a lot of these talks always feature some old picture of a very long dead European guy, and really we start in the 19th century Europe. This is the classification of diseases. Really comes from 19th century France. If you went back to the time of the American Revolution, a lot of the conditions that we're talking about today, nobody would really know what you're talking about. But if you brought this man, this is Duchenne, if you brought him to the present moment, he might know some of these things. And he would recognize a lot of the conditions that we're talking about. And I like to highlight him not just because he gives the name to Duchenne's muscular dystrophy, a very common disease that we see, but he's a whole lot more important than that. And he's not a guy who people have written a lot about. There aren't biographies of him. And he's really important for our specialty. So he invented these nerve conduction tests, which I'm sure some of you have had. And he was also the very first person to perform a muscle biopsy in a living human. And the technique that he used is actually still used. It's quite similar to what we do today. So that's the very beginning. And then we'll talk about what happens next. So over the subsequent 100 years after Duchenne and into the 20th century, really all of the neuromuscular disorders supported by the Muscular Dystrophy Association are described and classified. And at this time, we're in the middle of the 20th century. None of these have really any treatments to speak of. And partially because of this, the Muscular Dystrophy Association was founded in the United States in 1950. And then as we get into the latter half of the 20th century, there's this explosion in scientific information. The structure of DNA is fully discovered in 1953. And then over the next 20, 30 years, scientists get the techniques, the technologies available, and are able to discover the first genes for conditions that are known to be genetic. So genetic conditions are known for 100+ years before this, but we don't know what causes them until the 1980s, which is really when we start to see the discovery of some of these genes. 1983 is the discovery of the first gene that causes any disease, that's Huntington's disease, another neurologic disorder. In 1986, the Duchenne's muscular dystrophy gene, dystrophin, is discovered. And I should point out that a lot of the funding that got that done was actually from the Muscular Dystrophy Association. And then just a few years later, in 1995, you'll recognize that we're getting to a time that, a lot of us were alive during this time. These are genes that are really just discovered in the last 25, 30, 35 years. And I think that when we talk about, well, we finally discovered these genes, we have the technology, we can start to do things here. I think it's worth pointing out that we should pause and be a little cautious here, because throughout all of medicine, you can go way back, 200, 300, 500 years and hear of people talking about how we're going to cure this, we're going to cure that. And that should always give us a little bit of pause. And there's good examples for this in cancer treatment, heart disease. We say we're going to do something, and the promises that were made have not really been kept. And part of that is just that the science is not quite there in a lot of these conditions, and the technology isn't quite there. And despite the Nixon administration in the 60s and 70s saying, we're going to have a cure for cancer by the end of the decade, that hasn't happened. The Obama administration did the same thing. They made big advances, but the promises haven't held up. But I think I'm going to try to convince you that the current moment might be just a little bit different for some neuromuscular conditions. But I think that as we go into this, a little bit of skepticism can help inform the optimism that we have. And I think the way in which it can inform that is that we should be skeptical of marginal benefits. We should be skeptical of being told that the genetic technology can do so much and then getting a treatment that is only marginally beneficial or only beneficial for some people. What we really should be demanding now is home runs. And I'm going to show you that we have some proof of that we can do this, that for a lot of these conditions, we really should be striving for more. And then, in the 20th century, there's a couple of treatments, pharmaceutical treatments that come out for some of these disorders, riluzole for amyotrophic lateral sclerosis, Lou Gehrig's disease, steroids and Duchenne's dystrophy. And there's some benefits to these, but they really aren't, home run kind of benefits, life-changing, game-changing benefits. And the little slide I put here is from a internal medicine article about the utility of certain treatments and how much they cost, or tests and how much they cost. And this flat part of the curve you see on the upper right there, these are things that cost a ton of money and don't have a lot of benefit. And we really want to shift the curve down to the left here and have some treatments that are extremely effective, make a huge difference, and hopefully don't cost too much in the long run. And I think that we should pause here and talk about, well, how are we going to get these home runs, and really point out the role of the Muscular Dystrophy Association in this. And I said there's been a couple of treatments that have been approved, and there's been some more even over the last couple of years. And I think that we really should think of the Muscular Dystrophy Association and related groups as sort of, hit makers, people who are able to find the key features of, or the key scientists, the key people working in important ideas, and give them the support they need to get something done. And it's only been 35 years, not even 35 years, since the gene for Duchenne's dystrophy was discovered. And in that time, there's been several different approved therapeutics. There's been, $1 billion spent by the MDA and other groups. The number from the NIH is even more than this. And it's worth noting that in all of this, the Muscular Dystrophy Association is responsible for initial seed funding and other developmental funding for the majority of all approved treatments for neuromuscular disorders, especially neuromuscular genetic disorders. So this is a big deal. And with that, I think we'll run into a few specific examples. And the best place to start to tell this story, I think, is in spinal muscular atrophy. So spinal muscular atrophy is a common genetic motor neuron disease that typically affects infants and children. And when I was a medical student, which wasn't all that long ago, this is what we learned about spinal muscular atrophy. We were told, we had about 2 slides on it, we were told is a common genetic disease, the most common genetic cause of death in childhood, and that it's inherited in this way here, an autosomal recessive disorder, meaning that both parents have to be carriers and each of their children has a 25% chance of having both abnormal gene copies and having the disorder. But that's all we learned about it. That's it. But there's really a lot more to this. This is a fascinating, very interesting disease, a very rewarding patient population to work with. One of the early things we all learn about patients with spinal muscular atrophy is they're always described as having very bright eyes, very engaging. And you can even see this in the picture of one of the very first children described by a German physician, Hoffman, in the 1890s. And these kids are active, intelligent, bright, and really afflicted by a pretty severe motor neuron disorder. And like I said, this is a disease that all we learned about was that it's a common cause of genetic childhood death. And this isn't even that long ago. So a little more details on this. 95% of all patients with spinal muscular atrophy are missing or have mutated copies of a gene called SMN1. SMN stands for survival motor neuron. That's on chromosome 5. 1 in 50 people in the general public in the world are carriers for this condition. So if both people who are carriers have children, 25% of their children, every one of their children has a 25% chance of having this. And this leads to about 1 in 10,000 people in the world having this condition. And while we all learn it's a motor neuron disease, it's really much more than this. This is a multi-system disorder. It affects people's bone health, endocrine function, liver function, and much more than this. And it's, really the jumping off point for what we're going to talk about for what the future looks like. Now, the reason why this is maybe one of the first things that we start to get good treatments for is because in SMA, there's a second copy of a gene called SMN2. It provides very similar function, does basically the same thing as the other gene, and it can sort of cover for the lost SMN1 gene and protein product. And we've known now for about 20 years that the number of copies of this SMN2 gene that you have, the more likely it is that the more copies you have, the later your weakness will present and the more mild it will be. And here's a good example of that. So we, in previous iterations, broke spinal muscular atrophy up into types, type 1, 2, 3, 4, based on if you have it as an infant, a young child, an adolescent, etc. And you can see that the infantile form SMA1, children have on average 2 copies of SMN2, and as you get higher up, they have more and more copies. So this rescues the function of the lost gene product. So very astutely, as we learned this is not that long after this was discovered, people decided, well, if we could boost this SMN2 gene product, the protein here, maybe we could rescue some of this neuron loss and prevent neurons from degenerating. And what it turns out is this SMN2 gene has this extra, has this thing that when it's produced, it leaves out the 7th part of the protein, the 7th part of the genetic information called the exon. And when the 7th part is left out, the gene product doesn't do anything and it gets degraded. So technology that was partially funded by the Muscular Dystrophy Association, the small molecules goes in here, binds to this area, and makes the 7th exon get produced and produce more protein. So this starts to be done in the mid-2000s that people are doing this research. And in 2016, again, not that much later, this is only 21 years after the gene was first described, we get FDA approval for the very first drug to treat a deadly childhood genetic condition. So this is nusinersen, the brand name for this Spinraza. Several studies done that are published in 2016, 17, 18 demonstrate that there's pretty significant benefit to this drug. And it's especially true the earlier you treat patients with this. And here's some data from the initial paper in the New England Journal. This is the infant trial, where you see that compared to control patients, this is a trial that ended early because the difference was so big that they decided it was unethical to continue having control groups here. People do much better with this. And the earlier you do it, we think the better it is. And I think a good example of that is I took care of several patients with spinal muscular atrophy in fellowship back in Boston. And because of this, well, if we treat people early, what's going to happen? There were early studies, small studies done at Mass General and other places where people knew that their children had SMA or the child was diagnosed with SMA before he or she was even born and enrolled in treatment before they developed symptoms. So we had a family with 2 kids with spinal muscular atrophy. The older sibling was more affected, but being treated. And then we learned that the baby was affected before he was even born by doing genetic testing, amniocentesis genetic testing. And his family decided to enroll him in this trial and start him on treatment before he even developed any manifestations of weakness. And I came into his care about the time he was a year old. And a typical 1-year-old baby may be starting to take steps, is certainly crawling. And that's what he was doing. He's sitting, crawling, doing the things that a neurotypical 12-month-old will do. We saw him again at 18 months, and he is doing standard motor functions for 18 months old, walking quite well. I saw him again at 2 years, right at the end of my fellowship, and he's now able to basically run around. And because of this, the mentor I was working with actually had him tested again for spinal muscular atrophy because she didn't believe he had it. And there was nothing you could do to prove that he had this disorder. You couldn't tell. But when you did the genetic test, it turns out he really did still truly have it. So that sounds a little bit like a home run, doesn't it? Especially for that kid. But for others, that's not necessarily true. Like I said, we think the earlier you treat people before they've lost a lot of motor neurons, the better the benefit is. And it's important to note that Spinraza and nusinersen was approved based off of all of this data, which was really only in children, and randomized studies weren't done in adults. And you have to keep in mind that this is not the easiest drug to give. This is not a pill. This is a spinal fluid injection. You have to get a spinal tap every 4 months. And that has some risks associated with it. And I think it's useful to tell this story, but also point out, while it's very successful in certain instances, this is really kind of just the beginning of this technology. There's another modifier for this spinal muscular atrophy, cerebral motor neuron 2 protein gene in development. And this thing is called risdiplam. And many of you may have heard about this. The FDA is expected to come up with a decision for this at the end of the month on August 24th. This also boosts the SMN2 gene, but it's different in that it's a liquid, so you can drink it or use it in a G-tube, and allows more functional protein to be created, like I said. And this trial there's all these trials that have these cutesy names that are named after fish. Sunfish is the big one. And you'll note that, so this is a trial that has 231 patients. You'll note that it includes patients all the way up to 25 years of age. And the data that has been shared by the company is quite impressive, quite positive. But when top-line data is shared, we haven't had all of the ability to run through all of the data with a fine-tooth comb. The FDA certainly has this data, and that will help inform their decision on the 24th. But a lot of us who treat patients with spinal muscular atrophy, and a lot of patients are very excited about this possibility if it gets approved, because it's a different way to do the same kind of thing without the spinal tap every couple of months, which can be complicated, like I said. Now, spinal muscular atrophy has this separate protein product, the SMN2 gene, that can help boost this thing. Not all other genetic conditions have this, special out, so to speak. So in addition to trying to boost the SMN2 gene product, people have been trying to give the SMN1 gene back and actually treat the underlying problem. And There's another FDA-approved treatment for this. This was just approved a few years ago. This is gene therapy for SMA. It's approved for children under 2. It's a viral vector. So you are infected with a viral vector that delivers the SMN1 gene to your cells. It's a little risky. People get hepatitis, liver injury. A lot of that can be mitigated with steroids, but it's a single treatment. And in 2017, this data is published in the New England Journal, and you'll see cohort 1 receives a low dose of viral vector. Cohort 2 on the right-hand side receives a very high dose viral vector. And this is the CHOP intent score. So SMA patients will know what the scale is. It's a measure of all sorts of different activities that are a good marker of strength and function. And you'll see that all of these kids just really rocket up to quite a bit better than you would ever expect them to do without any treatment. So this is another big thing. And some of you may have seen Jerry Mendel's videos from Nationwide Children's that the MDA has helped produce about some of these kids who have been in these trials. So I think that to inform the rest of what we're talking about, it's worthwhile to mention how gene therapy works. Gene therapy has been around for 20, almost 30 years now. In the initial iterations, it was a little dangerous, and there were some complications with some of the early gene trials for other genetic conditions in the 90s. But we've gotten a lot smarter about this. So this is the way most gene therapies work now. I know a lot of us have been thinking about viruses for the last several months, but this is a good use of viruses. The virus is modified to include the new gene, so in the SMA case, the SMN1 gene, and then you give it to a patient and it gets into your cells, and a virus does what a virus does. It enters your cell, gets into your nucleus, and uses the machinery of your cells to produce genetic material. That's how viruses copy themselves. But we have placed into this virus a gene that we want it to produce, the SMN1 gene. And that's sort of how this works. So you could see most genetic conditions, you could try to do this kind of thing. So I hope what I've shown you is that spinal muscular atrophy is kind of the blueprint for where we're going with this. And this is spinal muscular atrophy is often talked about as the poster child for what you can do with genetic diseases and genetic technologies. And it's worth pointing out that it's only been 25 years since the discovery of what causes this disorder to having already 2 FDA-approved treatments and potentially 3 by the end of the month. They could really make a big difference in this disease. This is something that's completely unheard of. There's no other genetic condition for which this is possible yet. And I hope that this shows us that home runs are possible. We can do this. And it's not just because SMA sort of has a special biology or special genetics to it, but that we can do this with other things as well. So with that, we can talk about other conditions. And the next one that I think is worth mentioning is the Dystrophinopathies. This is what we now call Duchenne's and Becker's muscular dystrophy. This is one of the most common muscular dystrophies, one of the first described, affects boys, X-linked disorder. Women can be affected too, especially later in life. These are called manifesting carriers. Many of the advances here actually have a lot more to do with what we'll be talking about for the rest of the day with Dr. Distat and our colleagues in physical and occupational therapy. A lot of the advances that keep people functional and having good quality of life have much more to do with treating orthopedic issues, cardiac, respiratory issues. And this is really the foundation of what makes up a Muscular Dystrophy Association care center, an MDA clinic. Now, in the 90s and before, we started to learn that steroids have a pretty known impact on slowing the disease, blunting the disease somewhat. But anyone who's ever been on systemic steroids knows that these have a lot of side effects that can come up with them. So like I said earlier, these are beneficial, but not home runs yet. And hopefully I'll show you that we're starting to get there with some of this, newer genetic technology. And this is just a reminder of some of these things that give us a lot of benefit in patients with muscular dystrophies, nighttime splinting, mobility, power chairs to get you, able to move around as though, much better than you otherwise could. And then respiratory support with, respiratory technologies. So some of the newer therapies, we'll just detail a couple of little examples of what's going on here. The map that you see here to the right with all of these little numbers in it, this is simplified map of all of the different, what are called exons, the protein coding portions part of the dystrophin gene. SMA, spinal muscular atrophy, the SMN gene, there's only 8 exons. So it's a much smaller gene. The dystrophin gene is giant. It has 79 exons. And most patients with, if you're in an MDA clinic and you have 10 patients you're seeing with Duchenne's dystrophy, they're going to all have different mutations, all different genetic changes here. And that has a lot to do with, how they express their condition and when it develops and, other complications associated with it. But because of that, it makes this a little bit trickier of a condition to treat. But depending on the type of mutation you have, what's deleted or what's changed, you might be able to exploit this with some genetic technologies. And the first of this kind that's actually FDA approved is what's called exon skipping. So imagine you are a Duchenne's patient and you are missing exons 45 through 50. So if you cut out 45 through 50 there, and then 44 has to go connect to 51, and you'll see that the sharp end of 44 doesn't fit the sort of triangulated end of 51, meaning that when you try to put this together and make the gene product, it stops there and then it creates a non-functional protein. But if you go from 44 to 52, there's a nice connection there and you can create a protein that is only missing a few of the exons. And this is the theory for exon skipping. And the first one is exon 51 that's skipped. And, a sizable minority of patients with Duchenne's are actually amenable to using this technology. And this has been approved for a couple of years now. The real world data on how effective it is, can't speak much to that yet because it's, sort of an infancy in its use. But it's sort of a proof of concept that you really can do this and create more functional dystrophin protein. And then there's also a more recently in 2019, exon 53 skipping was approved, and that's conditionally approved. So they have to prove that it does something, that it's effective. And that data will be coming up in the next year or two. In addition, if you skip the mutated portion of dystrophin, you can create a slightly smaller form. You could also create a different type of dystrophin or dystrophin-like protein. There's another molecule called utrophin, which is very similar to dystrophin, but usually the body doesn't produce very much of it. So by boosting this, either with gene therapy, injecting things that make more copies of utrophin, or using small molecules that boost this response, the hope is that this will restore function. And in mouse models, this looks pretty promising. And we're starting to move towards human trials here too. And then the other types of gene therapy, where you just inject a gene in, are a little tricky to do with a giant protein, a giant gene product like dystrophin. But if you truncate it, you make smaller forms of it that still do the same job. These are called mini and micro dystrophins. The idea is that you can inject these in the same sort of viral vector and hopefully allow functional dystrophin to get done. A lot of these trials are getting set up right now, and these are sort of coming down the line here. So those are two of the major examples for Duchenne's dystrophy and Becker's dystrophy. And these are just some of the therapeutics, the genetic therapeutics. There's a lot more being done on different models of care, different technologies to improve people's function, improve their cardiac performance, improve their respiratory performance. And I think that when we get all wrapped up in the technology, we get really excited about this and can leave out some of the really important stuff that is very effective on everybody's day-to-day lives. So with that, we'll move to another set of conditions. These are the limb-girdle dystrophies. These are, unlike the prior ones that I talked about, these are 50 plus different conditions. We just still call them by this name, really only because limb-girdle dystrophies, the name came up to distinguish these types of muscle disorders from Duchenne's dystrophy, and then also from myotonic dystrophy, another thing, another condition that's quite common. So like I said, there's over 50 of these. Most of them, like SMA, are autosomal recessive. You have to have 2 mutated copies. And this classification system, LGMD1 is dominant, LGM2 is recessive. This is all changing. So next year we'll probably forget about this classification system. And the thing I really wanted to point out here is that I see a lot of patients who have what we call a limb girdle phenotype, meaning their shoulders are weak, their hips are weak, but their hands and forearms are strong, their face is strong, their feet are strong. I see a lot of patients like this, and no matter the genetic testing I do, I can't find a gene. I can't say, you have this particular type of limb girdle dystrophy. And in the published literature, this is about 40% of patients. So research strategies here really all depend on the specific gene mutation. There's a lot of different gene therapies that are under investigation by several different companies. The list here, 2A through 2E, and then 2I and 2L, these really represent almost the majority of patients with limb-girdle dystrophies. Limb-girdle 2A and limb-girdle 2B are the two most common that are around. And there's gene therapies that are under investigation that people are thinking about enrolling in clinical trials that are really about to start to get up and running for these. And there's early data from some of the 2C through 2E, what we call the sarcoglycanopathies, that the gene therapies are able to, restore function, at least on muscle biopsy samples. So a lot more to come on this. And then you'll note that as we've been talking about muscle diseases, I've left out myotonic dystrophy, which is a very complicated condition, a single gene condition, but there is a lot of research ongoing on this. And for those of you who have loved ones with myotonic dystrophy or who have it yourselves, I didn't leave it out for any good reason. Just we're limited a little bit on time here. The final condition I want to talk about is typically not a genetic condition. This is amyotrophic lateral sclerosis, or ALS, what we knew for most of the 20th century in the United States as Lou Gehrig's disease, after the baseball player who developed it in his playing career. This is an adult onset disease typically. Around 10% of ALS is genetic, but the rest of it, we think there may be genetic risk factors, but no specific gene that causes the particular disease. Similar to spinal muscular atrophy, this also leads to motor neuron degeneration, can really affect anything that requires muscle function. So chewing, swallowing, speaking, breathing, arm and leg function, rolling over. And unlike SMA, where I told you 1 in 10,000 people have it, the incidence statistic I don't think is very helpful, because this is sort of a rapidly progressive disease in some people. And I think the better statistic to show you just how common this is and how impactful it is that we do something about this is 1 in 400 people in the world actually die from ALS. And that may be an underestimate. And in the current year, there are really only 2 FDA-approved therapies. They're modestly effective, but there are a couple of therapies here. And I put a couple of pictures of famous people with ALS here, Stephen Hawking, Lou Gehrig, and then Gleason here, who has a big foundation. He was a football player who developed ALS in 2011 and has really dedicated his entire focus now to improving quality of life for patients with ALS, and also pushing research forward. So in addition to pushing some of the scientific, basic science research and clinical trials forward, he's really done a lot to get people the technologies and tools that they need to live the lives they want to live. And in ALS, there's really just an explosion of trials right now. There's always been a lot of trials in ALS. Over the last 20 years, there's probably been clinical trials. There's probably been right around 200. And of those, only 2 have really garnered FDA approval. That's riluzole and edaravone. But right now, there's just this huge explosion trying to do several different things to really get at what causes ALS, what causes the neuron degeneration, and ways to try and restore neurons that are lost. Many of you may have heard about stem cell trials. The biggest one is this Brainstorm trial run out of Mass General. That one, nobody really knows the results of yet, but the results are promised by the end of the year. And that's still true. So they think that they'll have them sometime in the fourth quarter. So certainly by December, we should know the answer here as to whether or not this stem cell approach they're using is working. And then you'll see there's several different things here. Masatinib is it's a tyrosine kinase inhibitor that affects the immune system. Cromolyn sodium, used for allergies and asthma previously, but thought to have some effect here. Complement inhibitors is another immune therapy. There's gene therapies for genetic forms of ALS. And then there's this platform trial, which I'll talk a little bit more about. And when I say explosion, I really mean explosion. So if you go to clinicaltrials.gov and look just for studies that are recruiting or about to recruit, not the ones that are currently done with and analyzing data, you get 169 studies. And a lot of these are treatment trials, not necessarily just outcome measures or natural history studies or other types of research that's done. So I said I'd talk a little more about this platform trial. So this is the NEALS and HEALEY Platform trial. NEALS is the New England ALS Association or consortium. It's a researchers that are based out of Mass General and the surrounding area. That they really are sort of the national research power horse for academic research in ALS. And HEALEY is the name of the ALS clinic at Mass General. A platform trial is very different than the typical types of clinical trials that we do, where we say we're going to study one drug in this patient population and see what happens. Platform trials are more common in cancer research, and this is actually one of the ways we've cracked the treatment for childhood leukemia and other types of lymphoma, for example, where you enroll patients in a trial, they all meet the same inclusion criteria, and then you can study multiple different things using the same outcome measures, the same research nurses, the same research facilities. You can get more people into the research, you can get more compounds studied, and there are some financial benefits. It's faster, it's cheaper to run a trial where you have sort of the same patient population and you try multiple different compounds in the same group of people. And this is really the first of its kind in all of neurology. And there's 54 sites around the country. These are just the initial 54 sites. They may expand this. A few of these sites are already enrolling. Many of them were expected to begin enrolling, and then the pandemic sort of put a damper on that. But some of them are starting to open for enrollment across the country. And you'll note that they're really kind of all over the country. Our region is a little bit limited in the Pacific Northwest. There's a site in Washington, a site here in Oregon. And then Idaho, Montana, Utah don't have anything, but most people who want to get into this trial can do so without having to travel great distances to do that. There's 54 sites. And like I said, there's 5 initial drugs that are being studied. They're going to start with 3 of them. And you'll see here on the top right that they're all sort of different compounds, immune modulatory drugs, nanocrystalline gold, several different mechanisms of action, really trying to see what works quickly and try and get it to patients faster. So with that, I think we can move into where we're going with all of this. I hope I've given you a nice tour of 4 different conditions and some of the successes and troubles that we're having. And I think where we're going really involves these key points. We have expanded genetic tools, expanded genetic treatments. We're getting better disease understanding from basic research. We're getting better ways to measure people's disease. And this is really a key point, which I'll come to again in a little bit. And the goal here with all of this is to develop treatments that improve quality of life, longevity, strength, and reduce the rate of all of the complications that we see in neuromuscular conditions. So like I said, there's a lot we still don't know. I mentioned that, especially for things like limb-girdle dystrophies, many people have a diagnosis, a presumptive diagnosis. We think you have a genetic muscle disease or a genetic nerve disease. This is also true in axonal Charcot-Marie-Tooth neuropathy, that many times you can't find the gene, at least with the currently available technology. And then, like I said previously, some of these conditions that are pretty well described, we don't have good, well-known ways to measure responses. So this has been a major issue with clinical trials in neuromuscular conditions, in that you want to try to test something, but the way we measure it hasn't been validated. So you try to validate it and you do a trial, and then the result either does or doesn't give you something that's useful. But these outcome measures are not fine-tuned. We don't know how well they, recapitulate people's function in the real world. And development of those, especially for the limb-girdle dystrophies, for FSH, for myotonic dystrophy, is really important, so that we know that the research we're doing, we know that the genetic information and technology we're taking from the laboratory can do something in patients, so we can measure the response effectively. For spinal muscular atrophy and ALS, there's really good validated scales to show us that our drugs work. But for a lot of these other conditions, we're still trying to develop these things. And we're doing these at the same time as we're developing the new therapeutics. So hopefully we'll be able to meet somewhere in the middle here. So ways to get closer to a diagnosis. If you or a loved one have a neuromuscular condition that is thought to be a genetic condition, but you don't know what it is, which is a pretty common thing, these are ways that you can get closer. So one way right off the bat, I would recommend that if you haven't already, go see a neuromuscular specialist at an MDA care center. There's over 100 of these care centers across the country, really all staffed by excellent people, very, very smart, dedicated teams of people. And sometimes all you need is that visit and that evaluation to say, oh, you must have this condition, let's send the test for it. But like I said, 40% of my limb-girdle patients, a lot of my neuropathy patients who have genetic neuropathies, we can't find the answer with the commercially available testing. And here I would point out this project, which is called the Rare Genomes Project. This is a project that's run out of Harvard and MIT. For the limb-girdle side of things, this has a lot of funding from the MDA and another foundation called the Jane Foundation. But it really is tailored to anybody with any presumed genetic disease where the genetic disease isn't fully described, the gene isn't fully identifiable. And you can go to this website, raregenomes.org, and there you can enroll yourself. You don't have to ask your doctor to enroll you. You can go and click through and see if you're interested in what they're trying to do. They'll collect, samples from you, and hopefully this will lead to a way to get a diagnosis for you. And I've done this for several patients so far. And so far, only one has come back because this is a process that takes a long time. But we're hoping that this is going to be a way to get closer to identifying more genetic disorders and ways to better identify ways to treat them. To understand treatments and outcomes, like I said, this is a big issue here. The MDA has had a database for patients for a while. It's really been updated and more used in a research fashion recently. So this is the MOVER data hub, the Neuromuscular Observational Research Data Hub. This includes 7 conditions now: ALS, spinal muscular atrophy, Duchenne's and Becker's, myotonic dystrophy, because these are diseases that have known outcome measures, but it's probably going to start to include other disorders in the future. And the real goals here are to improve outcome measures, improve best practices, which I know Dr. Distat is going to talk quite a bit about, how we actually care for patients in a way that's based in evidence, that helps people, that we know helps people. Improve clinical care for patients, and then also connect people to trials. This will include anonymized data of when you were diagnosed, what your genetic diagnosis is, all of these scales that we use in research and clinics. And it's a way to sort of stand up and be counted and know that there are, say in my clinic at OHSU, there are 56 patients with myotonic dystrophy. For example, and then we can say, well, now a therapeutic is coming up, we're going to be a site for a clinical trial. We have all of the data right off the top of our heads. Here's all 56 of our patients. Here's who qualifies for the trial. And that's a way to get people in faster without having to go through individual one by one, or patients having to lead the charge here and saying, I want to be in this trial. If we have all of the data in front of us, we hopefully will be able to get treatments out to people faster. And then we'll sort of get to the end here with, well, how do you find research? I think there's two good ways to find it. I'll tell you which one I think is the better way. So for neuromuscular conditions, I think that the Muscular Dystrophy Association website, Clinical Trial Finder, is much better option. It's much more fine-tuned. It lets you put in information about your condition or your loved one's condition, where you live, what you want to be involved in, and find these trials here. And then if you click this link on the website, there is also information about different aspects of clinical trials, different types of clinical trials, what you should know as a patient or a family member of a patient who's thinking about a clinical trial. And it really helps empower you to say, I do or I don't want to do this, while you're trying to find the trial at the same time. And then there's clinicaltrials.gov. This is a clearinghouse, federally run website. It's useful in that it is all inclusive. Every clinical trial that's run in the United States has to be on this website. The problem with that is that there are some trials that are run that are not worth your time, or are studied at one single center in the Florida Panhandle that you're never going to be able to get to, for example. And I think that using the approach on the MDA website is just a little bit more user-friendly, much easier to find things. So with that, we'll kind of wrap up here. We've talked about a lot of different conditions, and we have, of course, left many out. For the last 100, 200 years, these conditions have been very well studied, described, but there was really not a whole lot of progress in treatments, even in the quality of life treatments, the pulmonary treatments for some of these things, really until the latter half of the 20th century. I hope I've shown you that the genetic revolution, especially with spinal muscular atrophy, is beginning to return some dividends here. I think it's worth pointing out that a lot of what we're getting and why we're getting it is because patients, families, advocacy groups like the MDA, doctors, scientists, we've all gotten smarter, we understand things better, and we've all gotten more demanding. I don't think many of us want to sit back and say, we'll this therapy that does a little bit, we'll take it, and then we'll kind of sit back and rest on our laurels. We really want revolutionary treatments. And because of this advocacy, even without these new medications, and the work that everybody's doing, we're learning a lot more about how to treat and support patients with neuromuscular conditions. And a lot of this has to do with advocacy groups, patients and families standing up and saying that we want more. And I think that if we do that in terms of scientific development, we're going to get more dividends here too. And this is really, it's 2020. I talked about 5 approved treatments for SMA and Duchenne's dystrophy. This is the beginning. I'm hopeful that when we have iterations of this talk in 2022, 2025, 2030, that we're going to have to break it up even further and say, here's a talk about all of the treatments for Duchenne's and Becker's, here's myotonic dystrophy, here's facioscapular humeral dystrophy, et cetera. Because I'm hoping that we'll really get to the point that we've got treatments for these things that make big impacts for patients and families. And this, I really think, is only the beginning. And hopefully I've been able to convince you of some of that. And with that, I am happy to take any questions.
Thank you very much, Dr. Ragole. That was a great presentation. We did get a couple questions in. I know someone chatted in about their son having Becker's and where to register their DNA with Becker's. Would you have any suggestions for that person?
Yeah, I mean, I think that, and I know that Corey responded here, I think the Duchenne registry is great. And then, as this mover data hub gets up and running, I think that would be a great place to do that too, because the genetic information and the, and your patient reported outcomes and how strong you are and things like that all get reported and put in there. So when we get to the point where we have 20 people who have a similar nonsense stop codon, for example, then maybe we'll be able to say, well, here's what we can do with this. We have the technology to do this. And there's proof of this in concept. There's several patients with very rare genetic conditions who have been treated with one-of-a-kind genetic therapies at, Boston Children's has sort of pioneered this. But there's many people who are working on these kinds of things. And if you are at a place or know of a place where there are scientists working on this, you can get to this faster. And that's the hope of this data hub, is to get this information to people.
Exactly. We do have a question that was submitted. It is a person that was diagnosed with limb girdle back in '85. And they have recently, or I guess not recently, but they have learned that their dystrophy is caused by the mutation ADSSL1 gene. And two questions here. He's wondering where they could find more treatment options available. And I know that you talked about the clinical trials finder tool, but also, is this still considered a subtype of limb girdle?
That is a very good question. I gotta say that this is an extremely rare condition.
Okay.
Is it still? Well, I guess I don't think that you would call it a limb girdle dystrophy today. Back when you were first diagnosed, I think we would, because, like I said, limb girdle dystrophies were really everything that wasn't myotonic dystrophy and wasn't Duchenne or Becker's dystrophy. ADSSL, I think, is what we call a Nemelin myopathy. It's sort of a different thing. That's based on what the pathology looks like, or sort of grouped in what we call the congenital myopathies. But nowadays, these classifications, I don't think, are really all that useful, in the sense that, the limb girdle dystrophies get broken down into all these things, and there's different genetics to everything. And sometimes you really think someone has a certain genetic mutation, and then you do the genetic test and they have a different gene entirely. So really, I think that it's the gene that defines the disease. That's really how we should start to be thinking about this. And this is a pretty rare disorder that I don't know of any active, larger trials, but partially that's probably because how few books there are with it.
Okay. Are you familiar with VCP/PE97 disease?
Yes.
Okay. We have someone chatting in a question. He was just curious if you were familiar with that. So for you chatting in, yes, he is. Do you have a question relating to that disease? And then we have another question that came about. What about using and I'm probably going to not pronounce this right, acetylcysteine, CYS?
Oh yeah, N-acetylcysteine, yes.
Okay, does that help stabilize motor end plate in sarcoidosis and limb girdle?
Well, I think the jury is still out on that. I know folks are looking at this right now. There's a good sort of proof of concept that this is plausible, that you can do this. I don't think we know what the patient outcomes are going to be yet. But I think that this is another thing that's kind of in its infancy, that I'm hoping that in a few years we'll know a whole lot more about.
Okay. All right, we've got a couple coming in here. I have an 18-year-old with all, what is this? I have an 18.
They don't know which type of dystrophy.
Yeah, you can read that too. Are you referring to an 18-year-old or a 4-year-old? I'm a little confused with that. And let me just go to another one that maybe this person will type back in what the question is. This person has a son with limb girdle 21FKRP. What is the status of clinical trials of a sugar treatment? Are you aware of anything about that?
Yes. So there are, I'm sort of vaguely aware of this. The FKRP limb girdle dystrophy actually is now about to have gene therapy trials, which is yet another genetic therapy that we're hoping that can make a big difference in this particular disorder. But I know that there are ideas to try and give some of this product back. Some of that was done with some of my colleagues back in Boston. I don't know where that is in the trial development world, but I know that there's initial ideas on what it's going to do, how it's going to work, and how we're going to get it started up. But I don't know where it is in sort of the startup phases of these trials and where it's going to be involved yet.
Okay. I don't see any more questions coming in. I know that one person did not stipulate whether they had an 18-year-old or a 4-year-old or an 18-month. So I'm not really sure what that question is.
18-year-old
Oh,
son.
okay. 18-year-old son for 4 years serious testing for weakness in his lower legs. He's now in a chair.
I mean, I think what I would say to that, not knowing the whole clinical information, obviously, and thank you, Corey, for helping out with some of this. I mean, I really think that making certain that you get a good, fresh set of eyes is always important. And, sometimes patients tell me, hey, don't be offended, but I'm going to go see another doctor. And I really think that if there's questions about the treatment course or the diagnosis being offered, that no doctor should ever be offended by a patient saying that, because really we all want what's best for our patients. And if what's best for you is to go see somebody else and get a different opinion, I always wholeheartedly encourage that.
Mm-hmm.
And I think that's sort of the first step. And then if it's really thought, and Rosalie, I don't know the specific information about what condition it is that they think your son has, but if it's thought that this is a genetic condition, and there's been genetic testing done, I would consider looking at this Rare Genomes project and think about getting involved in there to see if they might be able to come up with a diagnosis. Because like I said, many of these conditions, we know they're genetic, but we can't find the gene. And even though we've gotten a lot further here, there's still a lot that's left, to uncover. And this is one way to get about doing that.
That's very true, very true. All right, so with the elephant in the room is COVID. I know all of us sitting here on this webinar have been highly acutely aware of what's going on. We do have a very valid question that's come in, and it says, how much risk is faced by MD patients going back to school? Should these kids go back to school? Should they not go back to school?
Yeah, I think that this is a really tough question, and a question that I honestly deal with every day in my clinic. And I think it depends on a lot of factors. And what I would say is that you really need to get good advice for your specific case, your loved one, your child, from their doctor about what they think the real risk is, because certain conditions are riskier than others. If you have spinal muscular atrophy and you are tracheostomy dependent, or you need to use bi-level pressure support most of the day, your risk profile is different than someone who has dysferlin limb-girdle dystrophy, where they have mostly foot drop, but their pulmonary function is well preserved. I mean, all of these conditions are so different, and we don't know enough yet about how this virus is going to impact the different conditions here. But what I think is really clear is that if there is respiratory involvement and reduced pulmonary or cardiac function, that these folks are at higher risk. And I think that is sort of the jumping off point to help make these decisions.
Very true. All right, thank you very much. We had someone type in, are you aware of any benefits of CoQ10?
I know it's been studied for several different conditions. I know that there are no harms to it, which I think is a good point to start. For certain conditions, there probably is some benefit. And, we talk about in the mitochondrial disorders that cause muscle weakness, that there are these, what we call mitochondrial cocktails out there that have CoQ10 and other enzyme sort of replacement things. There's not great evidence that those, make a, wild difference, make people stronger, make them have less complications. But the thought is that you're giving back something that otherwise you would have if you didn't have this certain condition. And I don't think there's any harm to it. But we just don't have the, like the robust evidence to say that this is, a game changer for people.
Sure, okay. We just got one more come in. Can you talk a little bit more about what you're seeing for myotonic dystrophy with any new trials on the horizon?
Yes, so myotonic dystrophy, so there's two types of myotonic dystrophy type 1 and type 2. They're very different conditions. Myotonic dystrophy type 1 is much more common. This is a very complicated genetic problem because it's a very large repeat expansion in an area of really genetic noise. And so what happens is that the genetic material gets kind of gummed up in the nucleus, and you can't produce the normal things you should produce, which is why it affects so many different things: muscles, eyes, breathing, heart. But there are some early studies looking at a molecule called CDK12. There's pretty good proof of concept. This just came out a couple of months ago. That helps relieve sort of this gumming up process in the nucleus. And the hope is that this can get into human trials pretty soon. And there's other approaches that are being done across the world to look at ways to try and fix this issue. There's some small molecules that people are using for congenital myotonic dystrophy to try and alleviate some of this genetic issue that I mentioned. But a lot of these are in their infancy, and I think that some of these are definitely not ready for prime time. The CDK12 therapy, I think probably if it gets to human trials, we're talking 5+ years. And then other things, there are some trials for congenital myotonic dystrophy for some of these compounds, but there's definitely more coming here.
All right, well, that's definitely promising news. I feel like there's a lot coming down the pipeline.
Yes.
We got to get the FDA to get in there. I do not see any more questions that have come up. So I believe that will probably be it, Dr. Ragole. Thank you very much. We definitely appreciate your time and all of your questions that were asked and answered. I hope that everybody got something that they were interested in. I just wanted to let you know too that up next we are going to hear about genetics from Corey Keller, and she was nice enough to type in some of those answers for us in the chat box. So we will continue back here at 12:15. So please stay tuned and feel free to chat in and connect with others. So thanks again, Dr. Ragole.
Of course, thank you.
Thank you.